Hello students, today we will start with new topic, that is variations from Mendel's principles. Variations that are studied during the inheritance, and those variations can be explained basing on the Mendel's principles. We know that variations means the changes. What are the changes? It can be due to sudden or during the course of evolution, whatever the changes that are observed. Now, these variations are observed based on the interaction of the genes, whether the genes are present on the same chromosome or between the genes that are present on different chromosomes. So, variations are due to interaction of genes. Here there are two possibilities. One is by genes in allele forms on same chromosome, or second possibility by genes in allele forms on different chromosomes. Here, whatever the interaction that is expressed by the genes can be studied. Now, these variations are of two types. So, before that, if you go for diagrammatic representation, if the chromosome is like this, here the allele forms of the genes, it can be present on the locus. So here on this locus of the same position of the gene, it can be the dominant or recessive allele, or here it can be a recessive or a dominant allele. So this can lead to, here I have represented about the genes in allele forms on same chromosome, for suppose. Now, variations, these are of two types, means interaction between the alleles is of two types. One is intragenic interaction or intra-allelic interaction. Intragenic interaction or intra-allelic interaction. Another type is second type of variations, or what type of interactions means. One is intragenic, second one intergenic interaction or interallelic interaction. Interallelic interaction. So here what are the examples that we can study under intragenic interaction or intraallelic interaction? Examples are incomplete dominance, that I will write clearly, separately. So here examples are: 1. Incomplete dominance. 2nd example, codominance. 3rd example, multiple allelism. Now, under intragenic gene interaction, we can study one more example, that is sickle cell anemia. Intragenic gene interaction. Example, sickle cell anemia. Sickle cell anemia. So, second type of interaction is So here examples are epistasis. Epistasis, one example. Another example is polygenic interaction. Polygenic interaction. Again, I am repeating. This type of interaction will be due to the genes in allele forms of same chromosome. This will be due to the interaction between the allele forms of the gene or gene in allele forms between two chromosomes. Now we will start with one by one, that is first incomplete dominance. When we are reading about law of dominance, I explained about one of the exception, that is incomplete dominance, because incomplete dominance is not getting expressed in all types of organisms. So we explained this one in the form of incomplete dominance. There are some exceptions I told. I also gave you two examples, that is Mirabilis jalapa and also Antirrhinum or dog flower or snapdragon flower. Now, since law of dominance is not getting expressed in all types of organism or all organisms, so it is considered as it is not universally applicable. That point also I explained in the previous class. Now, examples to explain this incomplete dominance are Mirabilis jalapa, which we commonly call as touch-me-not plant. Next one, Antirrhinum plant, which we also call as dog flower or snapdragon flower. Another example. These two are examples of plants or in plants. Now, another example in animals, that is Andalusian fowls. Andalusian fowls. Now we will take these all the three examples and we will explain this one in the form of Punnett square. That is ACE. So, this one we will directly take the parental allele forms or genotype and we will start the explanation. For suppose, in this case, either in Mirabilis jalapa or Antirrhinum. Parents are taken with the genotypes capital R. This is the genotype that is expressing dominant color of the petals of the flower, that is red color. It has red color petals. Now, when it is crossed with the recessive one, that is represented with small alphabets, we can also call it as red color flower also. Because mostly we will see the petal colors red in color, so that I have written here. This is considered as white color flower. White color flower. Now, when these both are crossed after obtaining of gametes, in F1 generation, we are seeing, if I draw the Punnett square of these two types of alleles. Now, capital R, dominance one I am representing on the top. Recessive characteristic alleles I am representing on the left side. Then we are getting heterozygous type of genotypes that are responsible for the third type of color, that is pink color. One dominant allele, one recessive allele, when they are side by side, they are getting mixed up now and they are expressing one of the third type of color, that is pink color flowers. Now, in F1 generation, all flowers are of pink color. Now, phenotypically they are pink in color. Genotypically they are heterozygous. If you write down a phenotypic ratio. Here, under phenotypic ratio, this capital R, small r means dominant one, recessive one. Combinedly, they are giving out third intermediate color. So, dominant is to recessive is to intermediate color. If you see here, dominant is 0, recessive is 0, but intermediate pink color are 4 in number. Next, here we can write here instead of this dominant or recessive, red color, white color also. Coming to genotypic ratio. Under genotypic ratio, if you write, all are heterozygous only. So if you write the parents like this, again it will be 0:4. Even in F1 generation also, the ratio of phenotypes and genotypes are same here in incomplete dominance. In the next F2 generation also, the both phenotype and genotypic expressions are giving same type of ratios. F1 generation offsprings are self-crossed. That we can represent as cross with capital R, small r. So these are the genotypes that we have obtained in the F1 generation. Now coming to F2 generation, the gametes are like this. Now in F2 generation, again we will represent this one in the form of Punnett square. So here in this F2 generation, we have seen again the homozygous red color, homozygous white color flowers. Now, according to Mendel's first law also, we have seen one of the explanation that the parental alleles sometimes in the F2 generation are not getting mixed up and expressing their phenotypic character, this one. But because in F2 generation, again we are seeing the parental types only. Here we will write the phenotypic ratio. Now we have to represent like red color flower, which is dominant. And in the F1 generation, we have obtained the third type of color, that is pink color flower, which is neither dominant, neither recessive, is to white color flower. Now, how many of these are number? Capital R means dominant homozygous alleles are representing only one red color, is to two pink color, is to one white color flower, which is purely a recessive type of color. Now, genotypic ratio. We can write the genotypes or allele forms of a gene is to homozygous is to heterozygous is to again recessive homozygous type. Now we have seen this one only 1 in number. Heterozygous forms are 2 in number. Again, homozygous recessive types are 1 in number. Here, if you observe, the both phenotypic ratio and genotypic ratio in incomplete dominance both are same. It means that incomplete dominance is showing both same type of ratios phenotypically and genotypically. Here, this is considered as the same type of phenotypic and genotypic ratio. Next, we will see about another example, that is about Andalusian fowls. Andalusian fowls. Now, your spelling is S, I think so. Andalusian fowls. If we explain this one in the form of the previous representation, like by drawing the Punnett square in F1 and F2, we will see what type of phenotypes and genotypes are being expressed. So first, if we take parents, let's represent the parents in form of capital B, which are responsible for the dominant color, that is black color feathers. Black color feathers. This is considered as the dominant one, dominant feature, which is crossed with the recessive one. This is showing phenotypically white color feathers, white colored feathers, which are considered as the recessive feature. Recessive feature. Now, Coming to the obtaining of a parental gametes. Parental gametes. It can be like this. So now, when we cross these two gametes, so in F1 generation, what are we getting? If we draw a Punnett square and check what type of genotypic expression and phenotypic expression are obtained. The allele arrangement is again heterozygous in case of the F1 generation offsprings. Now, capital B, small b means heterozygous combination of the alleles of genes are representing or expressing blue colored feathers. Blue colored feathers. So if we write the phenotypic expression of the offsprings in F1 generation. So phenotypic ratio. If we write like this, dominant is to recessive, it is not getting expressed. The dominant capital B, black color is not getting expressed. The third color is getting expressed. So it will be, now the third color, that is intermediate, that we are calling as blue color. Okay, that is 4 in number. So dominant is red in color, recessive is white in color. 4 intermediate color means the feathers of the fowls are blue in color. So this will be the phenotypic ratio. What about genotypic ratio? Genotypic ratio, in a sense, here, what are the types of allele forms that are seen in the offsprings? Now, here it is 0 in F1. This is completely all the offsprings. 4 means it doesn't mean only 4. How many number of offsprings it might be. So, again it will be 0. All are expressing this ratio of genotypes. So, now we will see about another type of example, that is codominance. Under codominance, if 2 different types of allele forms of a gene are present on an cell or something, then both of them will get expressed. One allele form or one gene is not dominating the other, but both of them, when they are side by side, they are independently expressing their own phenotypic nature. Now, phenotypes of both allele forms or of both genes are expressed simultaneously and no allele is dominating the other. Here, if we want to give an example, we can give the best example, that is AB blood group of the blood group type. So, one example is AB blood group. Now, we can also take another example, that is the coat color of the Jersey cows. For suppose if we recall the skin color of the Jersey cows, we see both some dark brown or black or color along with the white color patches. That is coat or skin color of Jersey cows. Now, another definition we also can give for this as codominance. Codominance is the expression of heterozygous alleles. Now, for this one, clearly we can take the skin or coat color of the Jersey cows. In a sense here, for suppose there are some dominant genes which are responsible for black color. Now, the same cow is also having the genes which are represented as like this, but these are also behaving as dominant alleles only. These are responsible for white color. Here it doesn't mean that it is recessive and it is dominant. Though it is means expressing one color and another type of genes are expressing another color, both are behaving as dominant alleles or dominant genes only. So that whenever we are writing here both alleles, we can represent like this. Okay, so we can also write like both allele forms behave like dominant forms only and get expressed. Now, clearly we will learn this example, coat or skin color of the cattle and also AB blood groups separately by taking out a Punnett square and representation. Example is skin or coat color of cows, Jersey cows separately, or we can write cat. Parents with allele forms or genotypes like this. No, gametes obtained are so this is showing the phenotypic black color. This is showing phenotype white color. Now, after the obtaining of gametes in F1 generation, if we draw the Punnett square, then the genotypes are like this. It doesn't mean, according to the topic, this capital B, which is considered as dominant in fowls, that is incomplete dominance. It is not like this. Here just we are taking one example for our codominance explanation and explaining that one. Here in F1 generation, both alleles express their own phenotypic trait, phenotype, as white and black patches side by side. If you want to explain this one in the diagrammatic view, see, think that this is an area of the cattle skin, for suppose. If you think this is white color, it will be having some black color patches here and there, like this. Like this. Okay, so this will be white color, which is getting expressed by this type of alleles, and this black color is getting expressed, is expressing its own color, and black color genotypic alleles we are taking like this in the form of capital alphabets. Now we can write this one as white patch, and this one also white patch. Okay. Now here all the offsprings are showing same type of phenotype. Even genotypically also here in F1 generation, the same type of capital B small b, means both the genes, whether they are in dominant form or recessive form, but both are getting expressed in the dominant form only. Representation can be similar to that of incomplete dominance, but phenotypically expression is different from that of incomplete dominant alleles in fowls, Andalusian fowls. Second example, that is AB blood group. on RBC of a person with AB blood group there will be both A and B antigens. Now, what are the genes or allele forms that are responsible for expression of such type of genes? Now, allele forms of a gene can be homozygous or can be heterozygous. In the same way, allele forms of blood group B gene can be homozygous or can be heterozygous. But now, when these both alleles are antigens are present on same RBC, then since both are of a dominant type, when they are side by side, both get expressed. Now, AB blood group person will not have any antibodies in his blood for these two types of antigens. Person does not have antibodies for this A and B antigens. In his blood. So, these are the two examples regarding codominance. One is coat or skin color of the cattle, that is Jersey cows, and another one is AB blood group.